Welcome to El Zoominario. Today we have Ariadna Gonzalez-Solis. She's a postdoctoral researcher at the University of Wisconsin-Madison. She got her PhD in biochemistry at the University of Nebraska-Lincoln and a master's in biochemistry from UNAM in Mexico. She's originally from Mexico City. Thank you very much, Ari, for joining us, and please go ahead.
Okay, thank you. Thank you so much, Claudia, for inviting me to give this talk. And thank you everyone for attending today to hear this story about plant membrane lipids. It's a tale of life and death, so I hope you enjoy it. But first, I would like to start just thanking the people that have been involved in all this research and also in mentoring me throughout my career. So especially thank you to Ed Cahun Lab at the University of Nebraska-Lincoln. That, as Claudia mentioned, that's where I did my PhD. So many people collaborated in this project. And also a special thank you to the lab of Marina Gavilanes in Mexico, where I did my master's and also my undergrad thesis. So thank you so much for all the people that have contributed into these projects. And so first, I would like to start by asking all of you a question. And is it what comes to your mind when you think about plant lipids, right? So if you think for a moment, what are you familiar with? What do you think first? And maybe some of you might be thinking about vegetable oils that are type of plant lipids. And basically, for example, here we have some pictures. And these vegetable oils, what they are mainly are triacylglycerols, and they form with a backbone here of glycerol, and they have attached 3 different fatty acids. So these are basically the molecules that form around 90% of the vegetable oils that you use for cooking, that you eat. And that's what most people think about the plant lipids. However, there's a lot more different plant lipids and that have different roles in a plant cell. For example, I'm pretty sure you all have seen the leaves, how that some leaves have a white cuticle, and that is epicuticular waxes. So some of those have different roles for the plants. So there's our surface protection, for example, against pathogens. They also have a role in trying not to let the plant lose a lot of water, depending on the conditions, the environmental conditions. We also have the different lipids that are forming membranes. So for example, as you know, plant cells have a plasma membrane, and they also have other organelles inside the cell, and they all have membranes, and they are formed by glycerolipids, phospholipids, and sterols. But moreover, there are other lipids that are also very important because they form chloroplasts and mitochondria, and that's where all the reactions, the electron transfer reactions happen. So for the plant to survive. Moreover, there are other lipids that can function as signaling molecules when the plant is coping or responding to different stresses. And the ones that I was just talking previously are these storage compounds that are represented here, like lipid bodies, where mainly the seeds use those as a storage compounds so that then the seed can germinate, and then from there you have a plant. So as you can see, there are different plant lipids, and they all have different roles in the cell. But today I want to talk about a specific class of lipids. These are my favorite plant lipids, are called Sphingolipids, and they have their name based when they first started to study them, they thought they were very enigmatic class of lipids that perform a little bit different to the other lipids that were being studied at the time. And that's what they named them as Sphingolipids, based on the Sphinx from Egypt. And so I've been studying these lipids now for a long time, and I find them fascinating. And most of them have different functions. So some of them, like you can see here in this cartoon, they are represented as this blue lipid here, and they are part of the plasma membrane, but they are also part of other membranes inside the cell, like the ER, and they get trafficked to Golgi. And they in that route, they also help proteins being trafficked from the ER to the plasma membrane. And then once in the plasma membrane, these sphingolipids also function as platforms for other proteins to organize and also have their function. They also can serve as signaling molecules. When the plant is responding to different stresses, they can also have that other function. So, and then, but how do they look like? Here I'm representing them just as a cartoon, but they are like a very diverse class of lipids, and they have different arrangement of structures. Here I'm representing the main 4 classes of sphingolipids. This, for example, is the basic, and this is kind of like where the synthesis starts. So then I'm going to highlight, as you can see here, they all have this long chain base or sphingosine base. This is like the trademark of sphingolipids. They all have this part. And then from there, they have another tail, another hydrophobic tail that is fatty acid. But then, as you can see here, they also vary and they have different head groups. So for example, this has a glucose, like a carbohydrate, but some others can get very complex, and that also affects how they are performing different functions in the cell. Studies that were done in Arabidopsis Taliana show that these sphingolipids that are more complex, the ones that have these very polar head groups, are highly represented in leaves, so around 64% this specific class, followed by this other one, 33%. And then these other classes of lipids, ceramides and long chain bases, are found typically in very low percentages in the cell. So as I was mentioning, for some of you probably are not familiar with Arabidopsis Taliana, and this is where some of the sphingolipid work has been done for plants. And this is a model system that we use because it is a small plant, and then it requires less space to grow. You can see here, for example, you can grow them in like an agar petri dish. So all these are small seedlings that you can use for doing experiments. You can also transfer these to small pots and you can do other experiments. And then you can also like propagate this plant and get the seeds. So you can have like large progenies for genetic analysis. The genome was sequenced a long time ago. So from then, there are a lot of genetic resources were available to generate mutants and to start studying this lipid class. So then, well, why are sphingolipids so important and why do we care studying them? So here I'm going to show you one example of why they are so important for and essential for plant development, right? So here on the left, we have an example of an Arabidopsis plant that it's a wild type. So this is kind of our reference plant that's growing under normal conditions of light, water, and without stress. You can see how this plant has this development. However, if we compare that to a mutant, this is like a sphingolipid mutant in which the synthesis has been affected. You can clearly see how now, compared this small plant to wild type, we have differences in development. And here where you have displayed all the leaves from this specific plant, you see how now the leaves are curled, and this is altered development. If you extract and do an analysis of the sphingolipids from wild type and compare them to the ones that you get from this mutant, surprisingly, the overall content on weight basis is the same, which means that the plants try to compensate not having a lot of lipids, just making the plant smaller. However, the composition is altered. But this is like an example of why affecting, just like having a reduction in the synthesis can have like a very, very high effect in these lipids. Another example here, it's a mutant, again from Arabidopsis thaliana, that in this case lacks the specific glucosylceramides, which are the complex lipids forming membranes. And here you can see a happy wild type just growing on agar, and you can start to see here the leaves. However, all these other images here correspond to the mutant of glucosylceramides, in which you can see here that the development will not carry over until these seeds are able to germinate, but then you won't be able to get a plant from here. You can see here how the true leaves cannot form from the seedlings. So this is just showing again, like how essential sphingolipids are in general for plant development. This means that if the plant cannot have these membrane lipids, then the plant cannot form. And then changing a little bit, I told you that the sphingolipids are very important for development, but then there's a lot of work also being done showing that sphingolipids are important for stresses. And I just wanted to highlight here, for example, all of you like have been visiting different places and have encountered like different environments, right? So here I'm just showing like 3 pictures. For example, this is from Lincoln this year, that we had a lot of snow and a very cold winter. If you compare these to the white sands in New Mexico or even in Wyoming, when you have like lakes and very different environmental conditions, you can see here in all of these different environments, you can have plants. And all these plants have to cope with the different environmental conditions. And that means that they have to sometimes adjust during the day and during the night to different conditions, and how that's gonna make them have to adjust depending on in order to survive. So as I was telling you, a lot of different stresses. So from drought, salinity, flooding, hot and heat, excess light, even pathogen attack, insect attack, and heavy metals or wounding, or just being the plants being damaged sometimes by the wind. All those are examples of stresses that if you think about it, the plant cannot just run, protect from the environmental conditions. So they have very sophisticated mechanisms to survive under the different conditions and adapt to. So another example that I want to share with you today is how the sphingolipids can be important to respond to the cold stress. So here again, we have Arabidopsis, our favorite plant. And before a cold treatment, if these plants, then you transfer them to 0 degrees centigrade for 4 weeks, you start to see differences, right? So these first 4 plants correspond to the wild type, which is our reference. And then the following plants correspond a mutant of the sphingolipid, specifically a delta 8 desaturase. And I'm gonna talk a little bit more about it in a minute. And then, but you can start to see how even 4 weeks have a detrimental effect in these plants. Other gene also that also encodes for the desaturase has a minor effect, but also you can start to see that the plants are not happy. Then if you combine those 2 genes and you have a double mutant of that specific desaturase, then you see how the plants are starting to be very not happy. If you then compare this for 8 weeks, now it's very clear how the mutant plants that don't have this gene cannot survive this cold treatment. However, and then when you complement, this means that if you put back the gene that corresponds to this function, then the plants behave like the reference, like wild type. And so this specific gene corresponds to an enzyme that can add, like this double bond, the difference that this enzyme is doing is just adding a double bond here in this position in a long chain base, in this sphingosine base. And only this change can have a huge effect on how these membranes can adapt to differences in temperature, right? Because what this double bond is doing is just increasing the membrane fluidity by adding this kick. So then all the membrane can adapt better to the differences in temperature. So now you all can go back, and when it's getting cold, when it's winter, you now can think about all the plants adapting to the stress by modifying their lipid in their membranes. Another example that we worked, I worked in that in the lab of Marina Gavilanes, was trying to see the sphingolipids at signaling molecules. And what we saw is that when you infiltrate just with a syringe plants with a pathogen and also with a toxin that affects the sphingolipid synthesis, you have basically the same response. This is just showing how the plants look like. These are leaves of Arabidopsis. However, if you also quantify different other signaling processes, for example, you see that the long chain bases accumulate, that the reactive oxygen species accumulate, and there's activation of the same kinases, and there's activation of gene expression. So this is an example in which this specific sphingolipid class can have an effect as signaling and can be a messenger telling the plant cells there's something going on. For example, in this case, a pathogen is attacking the cell, so we need to die in order to just contain the infection to this area and then let the rest of the plant die. And what now is very obvious is that sphingolipids are a second messenger in that pathway, in that signaling pathway. But then, okay, so I already told you that sphingolipids are very important for development and also very important responding to stimuli. So, but then how is a plant able to adjust to the different levels, and how is it even able to sense and then respond, right? So here I would like to, some of you probably are familiar with the Mexico City Metro, in which you can see different different lines that take you to different places. We all been there. We have all seen like this is like a flux of people trying to go from point A to point B. Sometimes it's very difficult to get in the metro. There's like a lot of people. On the other hand, the other direction is empty. So it's like a flux of people. And I always like to compare this with the biochemical pathways. So this is now on your right, there's like a simplified version of the sphingolipid biosynthesis pathway, where this is how it starts, but it's also like a flux. In this case, it's a flux of people trying to go from one point to the other. In this case, we have flux of different molecules that get converted at each different step. And then you have here like the final products. So the idea here is like, if we want to, for example, change we want to go faster from here to here, then we can increase, for example, this step, right? Or if we feel like there's a lack of the complex sphingolipids, we can the plant can adjust and try to make this synthesis go faster or go slower depending on the needs, right? So that is basically the regulation of the whole synthesis pathway. And it's very interesting here. The first step of the synthesis is performed by an enzyme, which is called SPT. And this is like the very first checkpoint, because if we are starting here, the flow of molecules that will give rise to the whole sphingolipid intermediates, then we can control and fine tune from here, and then we can either synthesize more or synthesize less. So this first checkpoint are proteins, and they are called the Orm proteins. So what we saw is that these Orm proteins are the ones that mediate the regulation of sphingolipids. And here are just represented the Orm proteins in red. And what happens is that when they come into contact with this enzyme SPT, the one that I told you that was the first enzyme in response to that synthesizes the sphingolipids, then they are negative regulators, right? So they stop the flow of the molecules trying to build up sphingolipids. And again, doing some experiments generating like a plant that has very high expression. So that means a lot of ORMs inhibiting these enzymes. Again, the plants are very, very small, right? There's not like enough complex sphingolipids to sustain the growth of this plant. However, what happens if now we have a mutant of ORMs? That means now we don't have the negative regulator. You would expect that probably you can have more sphingolipids, right? So this is exactly what I'm showing here. Again, this is like an agar plate growing different plants. These are the normal plants. However, you can see these little tiny plants that are the same ones that are shown here. They have a lot of developmental defects. They do not survive beyond the seedling stage. But if you can get a lot of these small seedlings and quantify the sphingolipids, you get the results like that I'm presenting in this graph. This is for wild type, but for these small seedlings, you can see how they accumulate up to 90-fold the amount of ceramides. So interestingly, most of the other sphingolipids can remain the same, but then ceramides are causing like this huge phenotype when the plant starts to build up a lot of sphingolipids. So with this, I would like to conclude the part of the research talk saying that plant sphingolipids, and this is the tale that I told you about the life and death, and it's all about balance, right? So as I told you, if plants have too little sphingolipids, they don't grow. But on the other hand, if they have too much sphingolipids, and that could be too much of a specific sphingolipids, then the plants develop cell death, or just the plants don't survive and cannot develop well. And so, but why is this important, right? So we know by now the pathway has been characterized a lot, and now what is important is like if we keep expanding this knowledge of how the sphingolipid metabolism works, and we also know more about how these different intermediates and how these different molecules can have an effect in the response of the plants to different stresses like pathogens or abiotic stresses, then that could improve tolerance to plant to the different the changes in the environment. So that's kind of like the overall goal. There's still many unanswered questions. Still, there's a lot of work going on trying to understand better how's the signaling pathway going on and how everything could be fine-tuned in order to have like plants that can tolerate better and can survive better. But this also is important because this can be translated to other studies and also in mammalian systems, in other systems. So it's very, very fascinating topics. And then I would like to briefly change and talk about a little bit of the culture. So as Claudia mentioned, I am from Mexico City, and I don't know if you have visited or been there. And so it's something that I'm very fascinated about Mexico City is that you have in the city center, like in downtown Mexico City, you can have like the big cathedral, and then, but then only like 2 blocks from there, you start going excavating, and then what you find is what was the ancient Tenochtitlan, right? So here, this picture represents like how the Aztec city would look like here, and they're surrounded by lakes, and then you have the mountains over here. This is how it looks like today. And something that I was very, very fascinated in 2006, they continued the excavations in Templo Mayor, and they were able to find like this monolith of one of the Aztec gods, and this is called Tlaltecutli. It was found there. There's still one piece missing that they haven't found yet, but also it's very interesting how all the colors, which are plant pigments, stay there, and they did a very good job at just going through the cleaning and trying to preserve all these pigments that are coming from plants and that have been stay there for long, long time. So if you haven't been in Mexico City, I would encourage you to go there and also look around and go to Templo Mayor. And also you can see this big monolith, which I think it's the biggest they found so far. It's like 15 tons, so it's huge. So yeah, I would extremely recommend to go. And then with that, I would like to just thank you for joining us today, and I would be happy to answer any questions.